So number 4, figure 4.1 shows 2 base pairs in a DNA molecule, shown here, and then A is name the bases labeled J and K and the bond labeled L. This is worth 3 points, and it's only asking you to name stuff. When this happens, each one of J, K, and L is worth 1 point. When that happens, that is what the breakdown is. Each one is worth 1 point. You don't even need to know that this is adenine. You don't even need that much information. That is free. Adenine is a purine. It has 2 rings. It has 2 hydrogen bonds with its complementary nitrogenous base. And because this is DNA, we know that this J must be thymine. It cannot be uracil because this is DNA. You do not need to know that uracil does not have a methyl group up here. That is not something you even need to memorize. You don't need to care. You can tell that this is thymine by the fact that this is DNA and that this is adenine. And that there are 2 hydrogen bonds means that they don't even need to tell you that this is adenine. There are 2 hydrogen bonds and this has 1 ring. That means it is thymine. It's DNA, 2 hydrogen bonds, 1 ring, thymine. It's the only possibility. K is a nitrogenous base. It is this one. Since they told you that this one is adenine, since they gave you that information, you can tell that these 2 are not the same molecule. The other purine is guanine. By process of elimination, this must be guanine. You don't even need to know. They don't even need to label that as adenine for you to figure that out. That's one way that you can figure it out. There are 3 hydrogen bonds here. And there are 2 rings. 2 rings means it's a purine. 3 hydrogen bonds means that it's either guanine or cytosine, and the purine is guanine. That means K can only be guanine. And they're asking you to name the bond. The L is pointing to the dotted line here. A dotted line is a hydrogen bond. That is 3 points. 3 free points. Because you don't need anything. You don't need to do any kind of real mental manipulation here. This is barely logic. You just need to remember guanine and adenine have 2 rings. Guanine to cytosine has 3 double bonds. Has 3 hydrogen bonds, excuse me. And adenine to thymine has 2 hydrogen bonds. And DNA has thymine instead of uracil. Uracil is in RNA. Those facts carry you through. You don't need to memorize chemical structure. You don't need to do any chemistry. You don't need to balance any equations. That's it. That's 3 points. That is 5% of the paper 2. 5% of the paper 2. That's the difference, actually, for most people, a letter grade. Yeah. 3 points. Now, here, they give you some information about penicillin, about the history of penicillin. It's an antibiotic that interferes with the synthesis of cell walls and bacteria, all the stuff that you may or may not remember or have learned. When they give you history, it really usually doesn't matter that much. But even before penicillin became widely available in the 1940s, the enzyme penicillinase, which breaks down penicillin, had been isolated. The enzyme is now found in many bacteria, and gives them resistance to penicillin. All that was just a preamble to the question. None of that is important for what they're going to ask you to do. Figure 4.1 is a ribbon model of a structure of the enzyme penicillinase. They tell you it's an enzyme, and they told you what penicillinase does in this. The arrow indicates the active site of the enzyme. They have an arrow pointing to the active site. Explain why the shape of the active site of an enzyme such as penicillinase is important. Do you need to know anything about penicillinase? No. Do you need any of the information they gave you, anything? No. They're asking you to describe active site. And it's worth 3 points. 5% of the paper too. To describe what an active site is. It's the place where a substrate binds. 1 point. It is specific to a substrate. 1 point. That an active site is where the chemical reaction occurs. 1 point. All of these things just describing the shape of an active site. If the active site shape is changed, a substrate cannot bind there. If an active site is blocked by an inhibitor, a substrate cannot bind there. Describing and explaining why the shape of the active site of an enzyme is important. Why do you need an active site? What does the active site do? What is an enzyme's active site? Define it, and what can you do with it? There's probably 6 or 7 ways to get those 3 points. Phrases that you put in, but they have to be coherent sentences. And for God's sake, you cannot use pronouns. Never, ever, ever, ever use pronouns. Because when you use pronouns, I have noticed something. You use pronouns to refer to multiple different subjects of the same sentence. It and it react together. Or this and this react. Which one is this? Which one is this? You'll use the same pronoun for 2 different things in the same sentence. I have no idea what you're talking about, and neither does the person grading your question. You must refer to the active site of penicillinase as the active site of penicillinase. You must refer to the substrate as the substrate. You must refer to the enzyme as the enzyme. You cannot use pronouns. Pronouns are dead to you. Never use a pronoun on the Cambridge exam again. If you start to write a pronoun, I want you to slap your wrist. It will lose you points because it is unclear. It is ambiguous. And ambiguity loses. Ambiguity loses. Because it will always be interpreted out of your favor, not in your favor. With reference to figure 4.1, identify the aspects of protein structure that are shown and those that are not shown. Are they asking you anything about the action of penicillinase now? No. They're still not asking you anything about what they talked about. They're asking you about protein structure. And these 2 paragraphs are also worth 3 points, 5% of the paper too. Aspects of the protein structure shown with reference to figure 4.1. What can you see here? You can see beta pleated sheet, alpha helix, and the whole thing is a 3-dimensional shape. So that means that you can see which levels of protein structure, secondary and tertiary. That is it. That is all you can see. Aspects of the protein structure shown. The aspects of the protein structure that can be seen in figure 4.1 are secondary structure as seen by the alpha helices and beta pleated sheet, and the tertiary structure as shown by the overall 3-dimensional conformation of the protein. That's 4 points right there. because I've described which two levels of structure, 1 point each, and I described how I saw them, 1 point each. Perhaps even by describing alpha helices and beta pleated sheets, depending on the rubric, that might even be 5 points. I might even be done at this point if the rubric doesn't specify that the first paragraph is limited to a maximum of a certain number of points. It might be. So the aspects of the protein structure that are not shown, what levels of protein structure are left? Primary and quaternary. Primary structure is not shown because the individual amino acids that make up the sequence of amino acids are not described. Quaternary structure is not shown because only one polypeptide strand, one polypeptide chain, is shown in figure 4.1. That's 4 points right there. I said primary structure, and I said quaternary structure. I described what primary structure was, and I described what quaternary structure was in reference to the figure. I always reference the figure because it specifies with reference to figure 4.1. If they say with reference to figure whatever, and you do not reference the figure, you cannot get maximum points. There is no way you will get maximum points. If you do not look at the picture and say, I see this, I do not see this, you will not get maximum points. That is the key to answering questions like this. You look at what is there and what is not there. You describe it, and then you define what you have described. So, in my explanation, I may have just earned 9 points, but I am capped at 3. How many sentences was that? 3 in this one, 2 in this one? Yeah, like 5 sentences total for 9 points that it's capped at 3. It does not take much to max out 5% of the Cambridge exam in 5 sentences. All it requires is a basic understanding of definitional knowledge of the levels of protein structure. And there is always some kind of protein question on the paper too. I have yet to see one where they don't ask a question about proteins somehow, some way. Figure 4.2 shows the changes in energy during the progress of an uncatalyzed reaction. Energy, peak, trough. Progress of the reaction going forward, figure 4.2. C. Draw on figure 4.2 a curve to show the change in energy during the progress of the same reaction when catalyzed by an enzyme. 2. That's worth 2 points, and they tell me to draw on the graph. I must draw on the graph, and the rules of the Cambridge paper 2 are for all drawings, I must use pencil. So I draw in pencil. If I drew in pen, and I make it ugly, or I mess up, I cannot fix that. I cannot ask for another test paper. Draw in pencil. Always draw in pencil, because no matter how confident you are, no matter whether or not you think you got it right, no matter if you know, if your hand wiggles, if your chair is wobbly, and your hand shakes, you can lose points for ugliness on the Cambridge exams, paper 2 and paper 3. Paper 1 is a bubble sheet, so who cares if your hand is ugly? But here, for a catalyzed reaction, the energy, it must start at the same place and end at the same place. That is probably the first of 2 points, because the catalyst does nothing to change the energy state of the reactants or products. It only lowers the activation energy. So I start at the initial point and the final point. And as long as my graph or my new line has a peak that is below this peak, I will likely get the second point. Starting at the same place and starting at the same place and ending at the same place is the first point, and having an activation energy that is lower than the original graph is the second. 2 points for memorizing that graph, preferably knowing what the heck you're doing. State the term given to the energy level that must be overcome before a reaction can progress. 1 point. That is 1 point for a definition. Those are the gimmies. Those are the easiest points available in paper 2. It is activation energy. You cannot call it activation. You cannot call it energy. You cannot call it activation quota. You have to call it activation energy. You must call it exactly what it is. Exactly what they want you to call it. If you do not, it is wrong. If you are given enough information to figure out more things, that is another part of the question. And finally, part D, by the way, is the first thing that might even vaguely relate to the information they gave you at the beginning. But that's Unit 5 stuff. So who cares right now? We haven't gone over it yet. Freezing temperatures can completely stop an enzyme activity by causing the molecules to undergo cold denaturation. Enzyme activity is not recovered when temperatures are increased to a normal working temperature range. They just defined something for you. An enzyme is denatured by cold instead of heat. Explain the mode of action of enzymes. Does that have anything to do with the definition they just gave you? No. But it's 3 points for understanding the mode of action of enzymes. It's always tested. The mode of action of enzymes is always tested. Whether by asking you about active sites, asking you about enzyme reactions, it's always tested. The mode of action of enzymes. 3 points for saying a substrate binds to the active site of an enzyme. It forms an enzyme substrate complex. The enzyme substrate complex undergoes a conformational shape change, which puts physical, mechanical pressure on the substrate, lowering the activation energy. And the reaction is allowed to proceed, and the enzyme releases the new products, and the enzyme active site is empty, allowing it to return to its original conformation. Maybe a 9th point. So yeah, there's like 9 more ways to get those 3 points. Understanding that statement, understanding that series, always gets you those points. Suggest how the molecular structure of the enzyme changes during cold denaturation. The three-dimensional conformation of the three-dimensional conformation, the tertiary structure of the enzyme, changes when the temperature raises and lowers. In denaturation, the change of the 3D conformation is beyond the point at which the enzyme can return to its original shape. And the 3D conformation is what allows an enzyme to function. If it cannot return to its original shape, the enzyme will cease to function entirely. I probably have enough here already. I could go on. I'm not going to because I'm tired right now. Cryopectants, cryoprotectants such as trehalose are of particular interest in their application to preserving cells, tissues, and organisms for future use. An investigation was carried out to find the protective effect given by different concentrations of two cryoprotectants, trehalose and glycerol, on a respiratory enzyme. The enzyme was suggested to freezing temperature and then returned to its optimum temperature. The activity of the enzyme was measured at its optimum temperature. Figure 4.2 is a graph showing the results of the investigation. Cryoprotectant concentration per millimole. The x's are trehalose and the targets are glycerol. And the percentage maximum activity is our y-axis, and that was our x-axis. So we have these two enzyme activity curves. As the cryoprotectant, which is the substance used to prevent the enzyme from denaturing, is as that concentration increases, the maximum activity increases in both. With reference to figure 4.2, describe the results of the investigation. I just did. Except when it says with reference to figure 4.2, describe the results of the investigation. With reference to figure 4.2. If they give you a graph and say with reference to figure 4.2, you better use the data at cryoprotectant at the trehalose concentration of 0, at concentration of 0 millimoles. Trehalose and glycerol percentage of maximum activity is equal at 0%. At 10 millimoles, the percentage activity of glycerol is 10% maximum activity, and of trehalose at 10 millimoles is 80% maximum activity. I'm using 2 data points. That's usually sufficient, but I'm going to go further. At 50 millimoles, both concentrations have reached a plateau, have begun to reach a plateau. With the glycerol concentration at 50 millimoles, the percentage of maximum activity is 90 something, whatever that corresponds to when you trace that number back. And it is 100% for trehalose. At 100%, at 100 millimolar concentration, they are equal at 100% of maximum activity. I have used 4 data points and described the data for each of glycerol and trehalose. I have probably gotten at least 2 to 3 points for that, because in the process of doing so, I have described the shape of both curves. I can also get additional points for saying that the difference is greatest between glycerol and trehalose at 10 millimoles, because glycerol has a slow increase in percentage maximum activity from 0 to 10, and it rises sharply from 10 to 50, and then tapers off from 50 to 100. And for trehalose, it rises sharply from 0 to 20, and then tapers off. If I describe the shape of the graphs, because I'm describing, I am not explaining anything. I explain nothing. I am merely reading the data and putting it into words, giving them the numbers themselves, and describing the shape of the graph. That's all I'm doing, and that's worth 4 points. If I needed to explain this, then I would simply say that at lower millimolar concentrations, trehalose provides greater percentage of maximum activity of as a cryoprotectant for the activity of the enzyme than glycerol. At high concentrations, millimolar concentrations of both cryoprotectants, the difference between them becomes negligible. So that is an explanation. Saying that one is better than the other is sort of beginning in the realm of explanation. It's sort of halfway between describe and explain. But also, if we were describing this in terms of enzyme activity, and these were two enzymes, you would say that this enzyme has a greater rate of reaction, or a more efficient active site, or a greater capacity, or a greater Vmax or something, or a greater substrate, or a greater enzyme affinity. More on that in a moment. So this is another one. This is kind of the same thing, but this is a state how the student could follow the progress of the reaction. And for that, the answer is always to measure the rate of change. You follow the rate of change. Explaining the results here. Explaining the results in figure 4.1, all the active sites are full. Concentration of catechol, the initial rate of reaction, concentrations of catechol, which catechol is enzyme catechol oxidase. So at that point, explaining this is all the active sites are full. So now we can get back to the subject at hand. This is how you will be tested. So now that we have seen what the substrate versus rate of reaction looks like, this is the rate of reaction versus time with two different concentrations of substrate and the same enzyme. The Vmax is the same here, or rather, the two different concentrations of enzymes, rather. The Vmax here is the same. The time from the start, from the start of the reaction, the rate of the reaction, and the time from the start of the reaction to the overall reaction completion. This is the course of the reaction over time. The higher the concentration, the faster the increase in the rate of reaction. The lower the concentration, the slower the increase in the rate of reaction. But the Vmax is the same for the same enzyme. Because when the active sites are all full, that's when you hit Vmax. So this is for the same substrate concentration. This is for the same enzyme concentration. My apologies. I apologize if in words it comes out much more garbled. So rate of reaction. Very quickly, now that it's in this, I can just say the rate of reaction is the amount of change over time. Change of the rate of change. So it's the change in the product over the change in time. That is your rate of reaction. Ideally, we're working in the initial rate of reaction. And we want instantaneous rate of reaction when time t equals 0. And that is from a homework assignment that I gave you then. I don't care about that now. Lab day, lab discussion, don't care. Follow-ups, don't care. Don't care. That was all. Wow. This is a lot of stuff. All right. So that was reminders. So now let's talk about enzyme kinetics. Enzyme kinetics are McCandless-Menten plots or curves or equations.
because of the scientists, because of the mathematicians and whatever that worked on it. So Vmax we're already familiar with. Vmax is the maximal velocity. It occurs at the point where all the active sites of the enzymes are full, where they are saturated with substrate. Saturated. They can hold no more. All enzyme active site has a substrate stuffed in it. So in this way, the Vmax is how fast can an enzyme catalyze a reaction. Km is a constant. It is graphically at the substrate concentration where at 1/2 Vmax. That is its location. It reflects the affinity of the enzyme for the substrate. The affinity is how readily an enzyme will bind a substrate. When an enzyme has a low Km, that is a high affinity. When an enzyme has a high Km, that is a low affinity. So Km does not affect Vmax. Km does not affect Vmax. That is just where it lives. That does not define it. This is the McCandless-Menten equation. The initial rate of reaction, V, is equal to Vmax times the substrate concentration over the McCandless-Menten constant plus substrate concentration. So, when the substrate concentration is so huge that it dwarfs Km, we can simply say that Km is negligible and we call it 0 for the sake of simplicity. When substrate concentration is enormous, it's so big that Km doesn't matter. It's the 0.1 to the 1 billion. It's the dollar, it's the penny that Bill Gates picks up. We can just call it 0 at that point. We don't care. That's when substrate concentration is so high. We just call Km 0 when substrate concentration is huge. At that point, it becomes Vmax times substrate concentration over substrate concentration. And I'm going to use the whiteboard for this. So we call Km 0. And it's Vmax times substrate concentration over substrate concentration. And we just use simple algebra now to just cancel stuff. Substrate concentration divided by substrate concentration is 1. And now our initial rate of reaction is Vmax. So when our substrate concentration is hugely high, ridiculously high, our initial rate of reaction is Vmax. Now, when substrate concentration is the same as Km, as the McCandless-Menten equation, then we simply replace substrate concentration with Km. And it becomes Vmax Km over Km plus Km. And that becomes Vmax Km over 2 Km. Km divided by Km is 1. And that means that the initial rate of reaction is 1/2 Vmax. Vmax over 2. That is the definition of Km. Km is the substrate concentration at 1/2 Vmax. That's the definition. That mathematically must happen. So, this is the McCandless-Menten plot. Vmax is an asymptote. An asymptote is the line where the line will continue going and approach that point, but never actually reach that point. and the asymptote is Vmax. 1/2 Vmax is the y value. Vmax is a y value. 1/2 Vmax is a y value. At the coordinate value, where y is 1/2 Vmax, the x must be Km. Km is a substrate concentration. It's a number. It's just a graph. It's just a graph. It's calculus with all the hard parts taken out. All the hard parts of calculus have been removed, but it is calculus. So, this was my original warning about how Cambridge does their slash thing for describing units. They use slash millimole instead of parentheses millimole to describe what the units are. So it's not divided by. It's in that unit. This is Km in millimolar units. Vmax in millimole per second units. This is how you read that. And probably also how you should write that on the paper 3. The slash whatever. Because they like that. It makes them feel warm and fuzzy inside. These are just some common examples. We don't care right now. We're done with that. Enzyme affinity is how readily enzyme substrate complexes will form. How readily an enzyme will react and bind to a substrate. E plus S, arrow with reverse, ES. How readily does that happen is enzyme affinity. A low Km will mean what for enzyme affinity? We've already gone over this. Low Km means high enzyme affinity. High Km means low enzyme affinity. And the substrate concentration needed to reach Vmax will be related to that. It depends on the amount of saturation required. So an enzyme with a low Km is more likely to reach saturation faster, and therefore will likely reach Vmax more quickly. And that way, it has a high enzyme affinity. If it reaches Vmax quickly, then that means that it will be, that means that its enzyme reaction, that its reaction rate will be less susceptible to changes in substrate concentration. After a certain point, the substrate concentration going up or down doesn't matter. After you've reached Vmax, it doesn't matter. So, where are my graphs of this? There we go. This line would continue all the way to Vmax. It would continue past this graph and all the way and hit Vmax eventually. So this is a low Km. This is a high Km. You can read the numbers and see that. Km is below 20. And here Km is above 20 millimoles. When the Km is low, it reaches Vmax more quickly. When the Km is high, it reaches Vmax much more slowly. It takes longer here. So the enzyme affinity is the inverse of Km. Low Km, high affinity. High Km, low affinity. This is them together. The lower Km reaches Vmax first. The higher Km reaches Vmax second. They will both reach Vmax eventually. If you continue raising the substrate concentration high enough, they will both reach Vmax eventually. But which one is more susceptible to changes in substrate concentration? This one. If you change the substrate concentration, the velocity, the rate of reaction will decrease. Lowering the substrate concentration lowers the rate of reaction. Raising it raises it. In the same area, it's not changing for the lower Km. You just look at the graph and see the difference. When you change the substrate concentration in this range, nothing happens in the low Km, and a lot happens toward the rate of reaction for the high Km. It changes a lot up and down for this one, not a lot up and down for this one. That is the effect of enzyme affinity and substrate concentration's effect on an enzyme reaction rate. So, now we have the factors that affect the enzyme affinity, the inhibitor concentration and regulatory